Hey guys, this is Dr. Keen, just talking to you about Chapter 3, Part 2, going through some cell transport information. I talked yesterday about all the different parts of the cell, and I told you we were going to talk about how that cell membrane works and how transport in and out of it works, and that's what this part is about. A little more complicated, a little more information about how that whole process happens, on whether something can get into a cell or not, and then how things get out of the cell, and that's all through a process called cell transport. So we talked about the cell membrane yesterday. It's got a double layer sheet of lipid, and we talked about those last chapter, the phospholipids that have a hydrophilic and hydrophobic barrier that holds the fluids of the cytoplasm and all the organelles inside the cell, and then it also on the outside allows for movement of the cell and for it to be cohesive with the cells around it. So it has that strong barrier. There's proteins that are embedded inside that as well, and we talked about those proteins a little bit when we talked about proteins last chapter. Those proteins actually create channels and pumps that allow for passage in and out of the cell. And then there's carbohydrate molecules that are attached to those proteins as well, and those can allow cells to identify each other. So they're kind of like markers that allow them to kind of determine what kind of cell they are, or where they're coming from, or whether they can be cohesive with each other or not. So here's an example of it. So these yellow dots here, that is the phosphate of the phospholipid, and that's the hydrophilic part. And then you have in here, these are the tails, or the fatty acids that come off of that phospholipid, and those create this hydrophobic environment here. Then you have these transmembrane proteins. It's called transmembrane because it goes across the membrane. And then you have the carbohydrate side chains off of here, and this is a protein as well. So these proteins in here, they're embedded in there so that you can, so that transport can occur. We're going to look at that in a second. And these little side chains here are for like recognition of this cell. So in order to talk about cell transport, we're going to talk about concentration. Concentration is the mass of solute in a given volume of solution, or mass per volume. So you've talked about this in other science classes, but and we talked about this earlier in the year, how solute is the molecule or the substance that's being dissolved in a solution, and then the solvent is what it dissolves in, right? So the mass of the solute per volume determines what the concentration of a solution is. So an example, 12 grams of salt in 3 liters of water. That concentration is 12 over 3, which is 4 grams per liter of salt in that solution. If you have 12 grams of salt in 6 liters, then you got 2 grams of salt per liter in that solution. So as you can see, the more solvent you have, the less the concentration is, because you have the same amount of solute in these two solutions, but this has less volume here than this does. So the more volume you have, the lower your concentration is going to be. So here's an example of how transport can occur. This is the simplest form of transport here. It's called diffusion, or when there's diffusion of water, it's called osmosis. And so here's water over here, and this is another solution with water and sugar in it. You can see these blue dots are water and the green ones are sugar. The concentration of water is higher on this side, on the left, than it is on the right. So with osmosis is naturally going to flow from a higher concentration to a lower concentration. The water is going to flow from the higher amount to the lower amount. And that's what's called diffusion. Like I said, osmosis is a form of diffusion. It's kind of a natural process that occurs just because of a difference in concentration. So, like I said a minute ago, it's trying to create some kind of equilibrium. It's trying to even those two solutions out. So it's moving solutes from the high concentration to the low concentration to try and create some equilibrium and balance it out. I'll let you go ahead and take time to watch this video on your own later. You have access to this PowerPoint on the assignment that I gave you today. So we're going to move past this and go into a little bit more information about diffusion here. Diffusion depends on particle movement and substances that diffuse across the membrane without requiring the cell to use energy. That's the biggest thing about diffusion, is there's no energy required for this to happen. Like I said earlier, it's kind of just a natural process. It just naturally flows from a high concentration to a low concentration. So that's something important for you to remember about diffusion, is that it does not require energy. It just happens. And it's just creating that equilibrium eventually and that balance eventually. And the reason why this happens is because particles are always in motion. They're always moving, and they're always trying to create some kind of stability. So that's why diffusion happens. So here's another example in a picture here. Here's some high concentrations over here, some lower concentrations over here. So it can move from high concentration to low concentration. Many substances can diffuse across biological membranes, but some are too large and too strongly charged to get through. So that if a substance can diffuse across a membrane, that's considered to be permeable. So that membrane is permeable to that substance if it's able to get through. If it's not able to get through, that's called impermeable. And like it said a minute ago, it can be because of its size, or because its charge, or because its markings are not correct for that cell. So that would be considered impermeable if it's not able to get into the cell. But most biological membranes, and I talked to you about this the other day, they're selectively permeable, which means they can determine, okay, you can come in, but you can't. And you're right for us, but you're not. So they basically determine what can come into the cell and what can't. And the way that they do that is with those proteins that we saw earlier and the markings to determine whether that's right or not. So again, osmosis is a type of diffusion. It's diffusion of water through selectively permeable membrane. So again, from high concentration to low concentration. So think about if you have a dam and it's blocking the water from going one place to the other. The water on one side is going to be higher than the other. It's going to be more water on one side than the other. But if you start getting holes in that dam, then the water's going to start pouring through those holes. And that is basically what osmosis is. It's that water flowing through that membrane and going to the other side from a high concentration to a low concentration. So in this example here, you got the blue is water and the orange is sugar. So you got a bunch of water over here, not so much water over here. A little bit of sugar over here, more sugar on the right. So if you're talking about osmosis, this is a partially permeable membrane, which means it can allow some things to go through and some not. So the water concentration is higher on the left than on the right, so the water is going to naturally flow to the right. So it's permeable to water, and so, but it's not permeable to the sugar. So it's flowing. The water's flowing from the left to the right to create that equilibrium. All right. So osmosis terms. Water will tend to move across the membrane to the left until equilibrium is reached. So when a concentration of two solutions is the same, that's considered isotonic. The word part iso means same. Tonic means concentration. So isotonic means same concentration and same on both sides. Hypotonic means that the solution one solution has a lesser concentration of solutes. If it's hypertonic, then the solutes are high. So if you're looking back at this one here, this one over here on the right, this is a hypertonic solution. This one over here is a hypotonic solution. So the water is flowing from the hypotonic solution to the hypertonic solution because of the difference in concentration. Osmotic pressure, for organisms to survive, they have to have a balance of intake and loss of water, and we've talked about that in class and how we need to replenish water. And in order to keep that normal osmotic pressure, we have to make sure that we have that equilibrium of water in our body, because otherwise we'll have hypertonic solutions in our body, and we don't want to have that. So osmosis exerts a pressure known as osmotic pressure on a hypertonic side, and a selectively permeable membrane, which allows for water to flow across the membrane to get fluids into an area that needs fluids. So that's the osmotic pressure. So the cell's filled with salts, sugars, proteins, other molecules. If it's placed in a hypertonic solution, then it's going to shrink, because if you put something in a hypertonic solution, it's going to suck all the water out of that cell because it needs the water. You put it in a hypotonic solution, then it's going to swell, because the water in the hypotonic solution is going to go into the cell because it's going into a higher concentration of water. So here's an example here. This is a hypotonic solution out here. You put that cell in there, all that fluid flows in to try and create some equilibrium. This is isotonic. They're both the same. If you put a cell in a hypertonic solution, it's going to want that water from here, so it's going to pull it out, and that's going to create a shrinking of the cell. It's trying to create that equilibrium. Facilitated diffusion is important to understand, because in this case, you're still going from a high concentration to a low concentration, but there's some help that's required. There's protein channels that are required. It's got to help that diffusion across, and so it's got to use these protein channels that we saw earlier. This occurs with charged molecules and molecules that are too large. So if there's a molecule that needs to get into the cell membrane and it's too large, it needs some help, those proteins will help open up a wider gate to allow those molecules to get in there. So net movement is still from a high concentration to a low concentration, and it still does not require energy. So you need to know that. Diffusion, osmosis, facilitated diffusion, they all do not require energy. Active transport is a different story, though. This is a transport that requires energy. You have to go from a low concentration to a high concentration. So this is going against the concentration gradient, which is what that's called. Going from a low concentration to a high concentration. So it's going against the concentration gradient, and look what we got. We got to use ATP for that, because it needs that energy to get that hydrogen ion out of the cell into a different solution. So this is another example, another schematic of how this works. So it's going against the concentration gradient. It needs energy, and it needs to transport proteins also. So just like facilitated diffusion, you got to use those transport proteins, but you're going the opposite direction, from low to high. And examples of those is endocytosis and exocytosis. Endocytosis is when a cell takes something into it, kind of like white blood cells. It takes something inside and it kills it. That's endo. Endo means inside. So it's taking something into the cell, and it requires energy and active transfer for that to happen. Exocytosis is the opposite. There's something in the cell that the body needs to get out, like with lysosomes, like waste products. So it's going to take it to the edge and it's going to spit it out. And that, again, needs energy and active transport using transport proteins. And that is part 2 of chapter 3.